Welcome to the deep dive. Today, we're really going deep, aiming for mastery of pharmacology's foundations.
That's right.
This isn't just, definitions. We're building that professional framework you absolutely need for rational therapeutics. We're looking at the language, the chemistry, the whole life cycle of medicines.
Exactly. This knowledge is, well, it's the bedrock. The goal here is simple. If you encounter any chemical intended for health, you need to instantly get where it fits, how it acts, and crucially, how its journey in the body dictates how you use it.
Okay.
We have to start with the core science, because honestly, confusion at this level, it just leads to poor decisions down the line.
Let's pin down the term itself, pharmacology. It comes from Greek, right? Pharmakon and logos.
Correct. Pharmakon meaning drug, or interestingly, poison. That duality is important.
Yeah.
And logos meaning discourse or science.
So the science of drugs and poisons.
Essentially, yes. It's the science dealing with how chemical molecules given from outside the body, the drugs interact with living systems. And our definition of a drug is pretty broad. Any single chemical substance that causes a biological response.
It covers a lot of ground then.
Oh, absolutely. The scope is huge. It's all the knowledge you need to use medicines effectively and just as critically safely.
It's interesting when you think about the history. For ages, it was all crude stuff, plant extracts, mixtures.
Where nobody knew the actual active chemical.
Right. And efficacy was pure guesswork, or maybe tradition. Getting from that to, well, science must have taken a huge conceptual leap.
It was revolutionary, and the key was borrowing methods from physiology. Before the 18th century, you might know what an herb did, generally, but never how. Then you get pioneers like Bejandy and Claude Bernard. They took animal experiments, originally designed just to study normal body function, and adapted them. They started to quantify, to analyze the specific effect of a specific chemical in a living system. That formal experimental approach, that was the essential first step toward understanding pharmacodynamics and kinetics.
And then it became official, institutionalized. Buchheim, Germany, 1847, the first Pharmacology Institute.
Exactly. That institutional support let the field really grow up. Which brings us to Oswald Schmiedeberg, late 19th century, often called the father of pharmacology.
Oh, okay.
His students, people like Langley working on receptors, Ehrlich on selective targeting, targeting the magic bullet idea, Abel isolating hormones. They laid down the fundamental concepts that took us from just using crude extracts to designing drugs targeted at specific molecules. It was a massive shift.
Okay, let's unpack the core structure then. You mentioned the whole field really splits into two critical concepts, two sides of the same coin almost.
They are absolutely the two pillars. You have to grasp both, pharmacodynamics and pharmacokinetics.
Right, PD and PK.
Couldn't have put it better. If you mix these up, you're lost from the start. Pharmacodynamics asks, what is the drug actually doing to the body?
And pharmacokinetics.
That asks the opposite. What is the body doing to the drug? It's the action and the reaction, fundamentally.
Let's tackle pharmacodynamics first, what the drug does to the body part.
Right. Pharmacodynamics from dynamics meaning power. It's about the drug's power, its effects. This covers all the physiological changes, the biochemical alterations the drug causes, and, importantly, its precise mechanism of action.
Mechanism of action, you mean how it actually works?
Exactly. How it works from the big picture, like effects on an organ system, right down to the nitty-gritty molecular level. Usually this involves interacting with specific biological targets, often receptors.
Okay, to make this really concrete, the sources give a great example, adrenaline. Its mechanism seems like a perfect illustration of this step-by-step effect.
It's a classic cascade, yes. The action kicks off when adrenaline binds specifically to its targets, the adrenoceptors. But that binding, that's just step one.
The first domino, like you said.
Precisely. Binding activates a G protein. Think of it as a molecular switch. This G protein then stimulates an enzyme embedded in the cell membrane called adenyl cyclase.
So the drug isn't the final actor, it's triggering a chain reaction inside the cell.
That's the key concept. Adenyl cyclase, once stimulated, ramps up the production of something called cyclic AMP, cyclic 3'5" AMP, technically inside the cell. This cyclic AMP acts as a secondary messenger.
And that's what causes the effects we see.
Yes, it's the rise in intracellular cyclic AMP that actually triggers the observable physiological effects, like the heart beating faster and stronger, that's cardiac stimulation, or the liver breaking down its stored glycogen.
Leading to higher blood sugar, hepatic glycogenolysis, and hyperglycemia.
Exactly. So pharmacodynamics maps out that entire sequence, from the initial molecular handshake, the binding, right through the internal signaling pathway, to the final measurable effect on the body. That's PD.
Okay, crystal clear. Now let's flip it. Pharmacokinetics, Kinesis movement. This is all about the drug's journey through the body.
Right. How the body handles the drug, its movement, how it gets changed.
Yeah.
And this whole journey is neatly summed up by that essential acronym, ADME.
Absorption, distribution, metabolism, or biotransformation, and excretion. ADME.
You got it. Absorption, distribution, binding or localization, biotransformation, and excretion. If PD explains if and how a drug works, PK explains how long it works and how much gets to the target site. It's about concentration and time.
Let's track a really common drug, paracetamol or acetaminophen, through its ADME profile. That should nail down these variables.
Paracetamol is a great example because it's generally very predictable kinetically. That predictability is a big part of why it's used so widely and safely. So you take it orally.
Absorption.
Absorption is fast and pretty much complete. It gets into the bloodstream quickly. You'll typically see the peak concentration in the blood within, say, 30 to 60 minutes.
That's quite fast. And distribution, where does it go?
It distributes quite widely and evenly throughout the body's water. The volume of distribution is roughly 1 liter per kilogram of body weight. This tells us it gets into tissues reasonably well, though it isn't super fatty, not highly lipophilic.
And binding, does it stick to proteins in the blood?
Minimally. Only about 25% binds to plasma proteins. This low binding is actually important. It means a large fraction of the drug is free, unbound, and immediately available to travel to where it needs to work, and also available to be processed by the body.
Okay, then biotransformation. Metabolism. This sounds like where the body really takes control.
It absolutely is. This is essentially the body's cleanup process. For paracetamol, almost all of this happens in the liver. The main process is called conjugation.
Conjugation.
Yeah, the liver chemically attaches large water-loving molecules, specifically glucuronide and sulfate groups, onto the paracetamol molecule. This makes the drug inactive.
Turns it off.
Exactly. And critically, it makes it much more water-soluble. These inactive water-soluble metabolites are then easily filtered out by the kidneys and eliminated from the body in the urine. That's excretion.
So the liver modifies it, the kidneys remove it?
Precisely. And this whole process, extensive metabolism, efficient excretion, is why paracetamol has a relatively short duration of action. Its plasma half-life, the time it takes for the concentration in the blood to drop by half, is only about 2 to 3 hours.
Which explains why you need to take doses fairly regularly to maintain pain relief.
Exactly. The kinetics dictate the dosing schedule. That's PK in action.
Okay, so we have PD and PK as the core pillars. But pharmacology doesn't exist in a vacuum. It connects to a bunch of other related fields, right?
Definitely. These related disciplines are where the pure science gets applied. Take pharmacotherapeutics. This is where you combine pharmacology knowledge with knowledge about a specific disease.
So using the drug knowledge to actually treat someone.
Right. It's the practical art and science of choosing the right drug, figuring out the optimal dose, deciding how long the treatment should last, and tailoring all of that to the individual patient's condition, their specific pathology, maybe their age, other illnesses. It's about preventing, mitigating, or curing disease using medicines.
Then there's clinical pharmacology. Sounds similar to therapeutics, but maybe more research-focused.
It is. Clinical pharmacology is specifically the scientific study of drugs, both old ones and brand new ones, in human subjects. Its job is to generate the hard evidence we need.
Like running clinical trials.
Exactly. Conducting pharmacokinetic and pharmacodynamic studies, first in healthy volunteers, then in patients. Running rigorous comparative trials to really pin down efficacy and safety versus existing treatments. And also, crucially, ongoing surveillance for adverse effects once a drug is on the market. The ultimate goal is to optimize drug use in people and provide the foundation for evidence-based medicine.
We touched on selective toxicity earlier. That's the domain of chemotherapy, isn't it?
Yes. Chemotherapy, in its broad sense, means treating systemic infections, bacteria, viruses, fungi, or cancers. It hinges entirely on that principle. Selective toxicity, finding drugs that harm or kill the invaders.
Bacteria, cancer cells.
With minimal damage to the patient's own healthy cells. This draws a really important conceptual line. You have pharmacodynamic agents like the adrenaline we discussed, where the main effect is on the recipient's body systems.
Right.
And then you have chemotherapeutic agents like antibiotics or antivirals, where the main goal is to inhibit or kill something foreign that has invaded the recipient. Different targets, different strategies.
What about actually handling the physical drug? Making the pills, mixing the liquids?
That falls under pharmacy and pharmaceutics. Pharmacy is the traditional art and science compounding, dispensing, preparing dosage forms. It also includes things like identifying raw materials, maybe some small-scale synthesis, ensuring quality and standardization.
And pharmaceutics.
Pharmaceutics is more the modern, large-scale industrial science. It's the technology of drug manufacturing. And, very importantly, designing the actual physical forms the drug comes in, the tablets, capsules, injections, patches we'll discuss later. Getting the formulation right is critical for the drug to work properly.
And the flip side of beneficial effects. That brings us to toxicology.
Right, the study of poisons. Toxicology looks at the harmful effects of chemicals, drugs included, but also industrial chemicals, environmental pollutants, you name it. The focus is on detecting poisons, preventing exposure, and treating poisonings.
It loops back to that pharmacon idea, drug or poison.
It absolutely does. Almost any drug can be a poison if the dose is too high. Toxicology reminds us that the line between therapy and harm is often just a matter of concentration. Understanding potential adverse drug effects is, fundamentally, a toxicological issue.
Okay, this framework is really solidifying. Now let's get into the actual substances. What are drugs, chemically speaking, and where do they even come from? This feels like where the rubber meets the road.
It really is. We move from concepts to the actual molecules. What are they made of? Chemically, most drugs are organic compounds. Think complex carbon-based structures.
But not all.
Not all. We still use some purely inorganic compounds, things like lithium carbonate for bipolar disorder or ferrous sulfate for iron deficiency anemia. Simple salts, basically.
And their properties? Acidic? Basic?
They vary. Some are weak acids, like aspirin. Others are weak bases, like morphine. Some are neutral non-electrolytes, like alcohol, ethanol. Physically, most are solids at room temperature that helps with making stable pills and capsules. But some are liquids, like ethanol again, or even gases, like nitrous oxide used in anesthesia.
No, one of the most critical parameters you mentioned, something that dictates a huge amount about how a drug behaves and how we can give it, molecular weight. The sources emphasize a key range, 100 to 1,000 Daltons. Why is that range so important?
This is absolutely fundamental to drug design and delivery. It's the Goldilocks zone for many drugs, especially those intended for oral use. Let's look at the boundaries. Why not smaller than 100 Daltons?
Yeah, what's wrong with being really small?
If a molecule is too small, it generally lacks the structural complexity needed for selectivity. To bind tightly and specifically to only its intended target, say, one particular type of receptor or enzyme, a drug needs a certain shape, size, maybe specific charged areas, perhaps chirality, meaning handedness.
Like a key needing specific cuts to fit only one lock.
Exactly. A very small, simple molecule is more like a crude bump key. It might interact with lots of different locks or biological molecules in the body, leading to widespread unwanted side effects. It lacks specificity.
So complexity enables selectivity, makes sense. What about the upper limit? Why not bigger than 1,000 Daltons?
That's mainly about getting around. It comes down to pharmacokinetics, specifically absorption and distribution. Large molecules, generally those over 1,000 Daltons, have a really hard time crossing biological membranes like the lining of the gut or the walls of blood vessels or getting into tissues. They're just too big and bulky to easily diffuse or be transported across these barriers.
So if you take a huge molecule by mouth, it probably won't even get into the bloodstream effectively.
For the most part, no. It won't be absorbed well, if at all. And even if it were injected, distributing out of the blood and into the target tissues could be a major challenge.
So that 100-1,000 Dalton range is the sweet spot for molecules that need to be specific and able to travel through the body, especially after oral administration.
Precisely. It's a key physicochemical conceit. Now, there are exceptions, of course. Lithium-ion is tiny, only 7 Daltons. And then you have biologics.
Like antibodies.
Exactly. Therapeutic proteins, monoclonal antibodies, these can be huge, 50,000 Daltons or much more. Heparin, an anticoagulant, is 10,000 to 20,000 Daltons. These large molecules completely break the rule.
And how do we handle them, then?
They absolutely must be given by injection parenterally.
Yeah.
They cannot be taken orally because they'd be digested or simply wouldn't be absorbed. The route of administration is dictated by their size.
Okay. And we also need to think about whether these chemicals are foreign to the body or not.
Right. Most drugs are technically venobiotic substances foreign to the organism, but a massive part of pharmacology involves using the body's own signaling molecules as drugs.
Like hormones.
Hormones, neurotransmitters, autocoids. These are endogenous chemicals. We can use them directly, like giving insulin for diabetes, or adrenaline in emergencies, or, very commonly, Chemists synthesize congeners.
Congeners, meaning related molecule.
Yes, molecular cousins. They take the structure of the natural substance and modify it slightly. The goal might be to make it more stable so it lasts longer, or more potent, or perhaps better absorbed, or maybe to reduce side effects. It's tweaking nature's design.
Okay, that covers the what. Now the where from. The sources list 6 main origins for drugs. Let's start with the oldest, plants.
Plants are the historical bedrock. For millennia, they were practically the only source. Much of our initial knowledge came from traditional medicine. Think about opium from poppies, digitalis from foxglove, quinine from cinchona bark, even things like curare used in arrow poisons.
And within plants, there are specific chemical classes we look for. Alkaloids seem key.
Alkaloids are hugely important. They're naturally occurring organic compounds, usually containing nitrogen, and they typically have alkaline basic properties. Many have profound physiological effects: morphine, atropine, quinine, codeine, caffeine. The list is long.
And they're often given as salts.
Yes, because as weak bases, they aren't very water-soluble in their pure form. Reacting them with an acid forms a salt like morphine sulfate or atropine hydrochloride, which dissolves much better in water, making them easier to formulate for injection or absorption.
But then there are glycosides. They involve sugar.
Correct. A glycoside has two parts: a sugar component, the glycone, chemically linked to a non-sugar active component, the aglycone. The sugar part often influences how the molecule is absorbed, distributed, or how long it lasts. The classic ones are the cardiac glycosides from foxglove, like digoxin, used for heart failure. The aglycone part is the active steroid that affects the heart muscle. Another group is the anthraquinone glycosides from plants like senna or cascara, used as laxatives.
What about oils from plants?
We need to be careful to distinguish here. You have fixed oils, which are chemically triglycerides, fats, essentially. Think olive oil, groundnut oil. They're non-volatile, have food value, and are often used as vehicles for other drugs or as emollients on the skin.
Okay.
Then you have essential oils. These are completely different. They are volatile, aromatic compounds, often terpenes, usually obtained by steam distillation like peppermint oil, clove oil, eucalyptus oil. They have no nutritional value. They're used mainly for flavoring in aromatherapy, or sometimes for mild local effects like counter-irritation or tormentative anti-gas effects.
And mineral oil.
Mineral oil, like liquid paraffin, is totally unrelated to plant oils. It's a petroleum product, a hydrocarbon, used as a lubricant laxative, sometimes. Important not to confuse them.
Got it. Other plant things?
Yes, things like tannins, which are astringent compounds used sometimes to treat diarrhea or locally on skin, and resins, gums used as demulcents or suspending agents. And we can't forget relatively recent discoveries like artemisinin from Chinese wormwood, a vital antimalarial drug, which is a sesquiterpene and doperoxide, a unique chemical structure.
Okay, moving from plants, animals were another early source, right?
Yes, particularly in the late 19th and early 20th centuries, before we could synthesize things easily. Extracting active substances from animal organs was a major breakthrough. This gave us hormones like adrenaline from adrenal glands, thyroxine from thyroid, and crucially, early insulin preparations from pancreas. Also, things like antisera, containing antibodies, and vaccines were derived from animal sources.
Next source, microbes. Hugely important.
Immense. Microorganisms, mainly fungi and bacteria, are the source of the vast majority of our antibiotics. Penicillin from Penicillium fungus is the classic example, but also streptomycin, tetracycline, erythromycin, gentamicin. Most major classes originated from microbes. They also give us some important enzymes, like streptokinase from streptocarci bacteria, used to dissolve blood clots.
What about minerals? Are many drugs minerals?
Relatively few drugs are used directly in their simple inorganic mineral form, but they are important ones. Iron salts, like ferrous sulfate for anemia, lithium carbonate for bipolar disorder, magnesium or aluminium hydroxide as antacids, zinc oxide in skin preparations. So not a huge number, but some key players.
Which brings us to the giant source today, synthetic chemistry.
This is now by far the largest source of new drugs. It really took off in the 19th century and revolutionized medicine. The advantages are massive.
Like what?
Purity, you get exactly the molecule you want, nothing else. Uniformity, every batch is identical. Quantity, you can make as much as needed. You're not limited by harvesting plants or animals. And crucially, design.
You mean creating totally new molecules?
Exactly. Chemists can create congeners, those modified versions of natural products, maybe making a semi-synthetic penicillin that resists bacterial enzymes. Or they can invent entirely new chemical families that have no natural counterpart, like the benzodiazepines, Valium-like drugs, or thiazide diuretics.
And modern synthesis is even more targeted.
Oh, absolutely. A lot of modern drug discovery involves rational design. You identify a specific biological target, say an enzyme crucial for a virus, or a receptor involved in high blood pressure. Then chemists design and synthesize molecules specifically shaped to interact with that target. Think of ACE inhibitors designed to block the angiotensin-converting enzyme, or drugs targeting HIV reverse transcriptase.
Synthesis also helps with chirality, right? The handedness issue.
Yes, that's a major advantage. Many organic molecules are chiral. They exist as non-superimposable mirror images, like your left and right hands. Often, only one hand, an enantiomer, has the desired therapeutic activity, while the other might be inactive or even cause side effects. Chemical synthesis allows us to produce just the single active enantiomer, leading to cleaner, sometimes more effective drugs.
Okay, and the final, most recent source, biotechnology, recombinant DNA.
This is the cutting edge, particularly for producing large, complex, protein-based drugs. It tackles that molecular weight problem we discussed. We use genetic engineering techniques inserting human genes into bacteria, yeast, or mammalian cells to make them produce human proteins.
Like human insulin.
Exactly. Human insulin, human growth hormone, interferons, erythropoietin, which stimulates red blood cell production. These are all made using recombinant DNA technology. It allows us to produce complex biological molecules reliably and in large quantities.
And this field is still growing fast.
Immensely. The newer frontiers are things like monoclonal antibodies, highly specific antibodies designed to target cancer cells or inflammatory mediators, and various growth factors. Protein therapeutics is a huge and rapidly expanding area of pharmacology, but again, remember size constraint, these almost always require injection.
Okay, that gives us a fantastic overview of what drugs are and where they come from. Now, if we have all these powerful substances, managing them, naming them, standardizing them, controlling their use must be incredibly important. The logistics and regulations seem critical.
They are absolutely crucial. You have potentially thousands of active chemicals. If you don't have clear, unambiguous systems for naming, defining standards, and regulating access, you have chaos. And in medicine, chaos means patient harm.
So let's start with the names. Every drug seems to have multiple names, which can be confusing. The sources break it down into three main types. First, the chemical name.
The chemical name is the precise, unambiguous description based on the rules of chemical nomenclature, like that example for propranolol. 1-isopropylamino, 3,1-naphthoxy, propan-2-ol.
Nobody's writing that on a prescription.
Absolutely not. It's completely impractical for everyday use. It's essential for chemists and regulators to define the substance, but not for clinicians. Before a drug even gets a proper name, manufacturers often use temporary code names or numbers, like RO151788 for what later became flumazenil. These are just internal identifiers during development.
So the practical name we need is the non-proprietary name.
Correct. This is the official name, if you like. It's assigned or approved by designated national bodies, like the U.S. Hand Council in the U.S. or the BAN in the U.K., and internationally coordinated, ideally, by the World Health Organization. The WHO promotes the use of recommended international non-proprietary names, RINNs, to try and get global uniformity.
So everyone calls the same drug by the same non-brand name.
That's the goal. And once that name appears in an official drug reference book, a pharmacopoeia, it technically becomes the official name.
Okay, but here's where the common language gets tricky. Everyone calls this the generic name, but the source material says that's technically wrong.
Etymologically, yes, it's a bit of a misnomer. And it's a really important distinction for true mastery. Generic should really refer to the class or group the drug belongs to, the genus. Like penicillins is a generic term for that whole family of antibiotics, or phenothiazines for a class of antipsychotics.
But in practice?
In practice, almost everyone, doctors, pharmacists, patients, regulators, insurance companies, uses the term generic name to mean the non-proprietary name, the official chemical name minus the brand. For example, atorvastatin is commonly called the generic name for the drug marketed under the brand name Lipitor. While technically imprecise, you just have to accept that this is the common usage.
Okay, understand the distinction, but use the common term. The third name, then, is the proprietary or brand name.
Right. This is the name owned by the pharmaceutical company that markets the drug. It's their trademark. These names are chosen for marketing reasons. They're often designed to be catchy, easy to remember, maybe hint at the drug's use. Think Viagra, Prozac, Lipitor.
And one drug can have many brand names.
Oh, absolutely. The same non-proprietary drug, say amlodipine, might be sold by different companies under dozens of different brand names worldwide: Norvasc, Istin, Amlocor, Stamlo, and so on. This can obviously create confusion.
Which leads directly to the big debate. Should doctors prescribe using the generic, non-proprietary name or the brand name? The arguments for generic seem strong uniformity, cost savings, maybe better understanding if names share a stem, like OLOL for beta blockers.
Those are definitely the strong points for non-proprietary prescribing. It promotes clarity, helps in recognizing drug classes, and often significantly, it's cheaper for the patient or the healthcare system because generic versions are usually less expensive once the original patent expires.
But there's a counter-argument, especially concerning quality.
There is, and it's crucial, particularly in situations where regulatory oversight might be less rigorous. The argument against always prescribing generically hinges on bioequivalence.
Bioequivalence, meaning the generic performs identically to the original brand.
Exactly. While the active chemical must be the same, the formulation, the inactive ingredients, the manufacturing process can differ between the brand name product and various generic versions. In well-regulated markets, generics must prove they are bioequivalent. They have to deliver the same amount of active drug to the bloodstream over the same time period as the original.
But if that proof isn't reliable,
Then there's a potential risk. A poorly made generic might release the drug too slowly, leading to treatment failure, or too quickly, potentially causing side effects or even toxicity. In such environments, some clinicians argue that sticking with a specific brand name product from a manufacturer they trust provides an extra layer of assurance about consistency and reliable bioavailability.
so the choice isn't just about cost, it's also about confidence in the manufacturing and regulatory standards.
Precisely. It becomes a risk-benefit assessment based on the specific drug, the patient, and the perceived reliability of the available products. True mastery requires understanding this nuance, not just defaulting to the cheapest option blindly.
Okay. To navigate all these names and drugs, we rely on reference books compendia. You mentioned official versus non-official.
Yes. Official compendia are those recognized by law, setting legal standards. Non-official compendia are highly valuable reference works, but don't carry that same legal weight.
The main official ones are the pharmacopoeias.
Right, like the United States Pharmacopoeia USP, the British Pharmacopoeia BP, the Indian Pharmacopoeia IP. These are the definitive technical references. They contain detailed information on chemical structure, physical properties, methods for chemical analysis, assays, standards for purity, required storage conditions, and lists of approved dosage forms for each drug.
Sounds essential for manufacturers and regulators.
Absolutely. They ensure quality control. But they're generally not very useful for practicing doctors because they lack clinical information. No doses, indications, side effects, etc. It's all technical specs.
For the clinical information, we need formularies.
Exactly. Formularies like the British National Formulary, BNF, or the National Formulary of India, NFI, are designed for prescribers. They're usually compact booklets listing selected drugs available in that country or healthcare system. They provide the practical info needed at the point of care. Indications, dosages, available forms, tablets, syrups, etc. Contraindications, important side effects, maybe drug interactions. They're often organized by therapeutic class, making them easy to use.
Is there a difference between, say, the BNF and NFI?
A key difference mentioned is that the BNF includes brand names and often cost information, making it useful for navigating the specific market. The NFI, historically, focused purely on the non-proprietary names and clinical data, without brand names or costs.
And for a truly comprehensive global reference.
The gold standard non-official compendium is generally considered to be Martindale, the complete drug reference. It's an exhaustive, incredibly detailed compilation of information on drugs used worldwide. It covers pharmaceutical aspects, pharmacology, therapeutic uses, international names. It's an unbiased, deeply researched resource, updated regularly, indispensable for complex drug information needs.
Okay, beyond naming and references, there's regulation of access. Prescription versus over-the-counter seems like the main divide.
It is the primary one for patient access. Prescription drugs, like Schedule H drugs in India, require authorization from a licensed medical practitioner. This is usually because the drug is potent, has significant potential for harm if misused, requires monitoring, is potentially habit-forming, or treats conditions requiring a proper diagnosis. Most drugs, including all antibiotics, fall into this category.
You mean OTC?
Non-prescription or over-the-counter OTC drugs are those deemed safe enough for the public to select and use themselves for minor ailments, following the label instructions. Things like simple painkillers, some strengths, antacids, cough drops, some antihistamines. The key assumption is they have a wide safety margin when used appropriately.
There's also a special category, orphan drugs. What defines them?
This category was created to address a market failure. An orphan drug, as defined initially by the U.S. Orphan Drug Act of 1983, is a drug intended to treat a rare disease typically defined as affecting fewer than 200,000 people in the U.S. Or, interestingly, it could be for a common disease that is only prevalent in resource-poor countries.
Why do they need special status?
Because the potential market is so small, pharmaceutical companies often couldn't expect to recoup their massive research and development costs through sales. There was no financial incentive to develop drugs for these orphan conditions.
So government stepped in.
Yes. Laws like the Orphan Drug Act provide significant incentives, things like tax credits for R&D, grants, extended market exclusivity periods after approval, to encourage companies to develop treatments for these rare but often devastating diseases. Drugs like imatinib for CML or enzyme replacement therapies for rare genetic disorders exist because of this legislation. Even some older drugs like lithium or colchicine have gained orphan status for specific rare indications.
Moving from rare diseases to global health policy, there's the WHO's essential medicines concept. What's the core idea here?
It's a profoundly important public health strategy, first launched by the WHO in 1977. The core idea is to identify a limited list of medicines that are considered essential to meet the priority healthcare needs of a population.
Priority needs, meaning the most common or serious conditions.
Exactly. The selection isn't based on what's newest or most profitable, but on public health relevance, proven efficacy, acceptable safety, and crucially, comparative cost-effectiveness.
So it's about getting the most health impact for the available resources.
Precisely. The ultimate goal is ambitious: to ensure that these essential medicines are available within the health system at all times, in adequate amounts, in the appropriate dosage forms, with assured quality, and at a price that individuals and the community can actually afford. It's about equity and rational resource allocation.
The selection criteria must be incredibly strict then.
They are, and they're multifaceted. First, there must be solid evidence of efficacy and safety from clinical studies. Second, the quality and stability of the product must be guaranteed, even in challenging climates or storage conditions. Third, the choice must reflect the local burden of disease, what are the most common and serious health problems in that region, and consider the available resources and infrastructure.
And the comparison aspect is key.
Vital. When choosing between several drugs for the same condition, the decision should be based on a careful comparison of their relative efficacy, safety profiles, and cost. Value for money is a major factor.
What about combination pills?
The WHO generally prefers single-compound medicines. They're usually simpler to manage, dose, and procure. Fixed-dose combinations, FDCs, where 2 or more active drugs are in one pill, are only included on the essential medicines list if there's clear evidence they offer a distinct advantage. Maybe better patient adherence, a synergistic therapeutic effect, or sometimes helping to prevent the development of drug resistance, like in TB or HIV treatment.
And this list isn't static.
No, it's a continuous review process. New evidence emerges, disease patterns change, new drugs become available. The WHO model list is updated regularly. The 20th edition from 2017 had 433 medicines. Countries then adapt this model list to create their own national lists of essential medicines, like India's 2015 list with 376 drugs, often specifying which level of care, primary, secondary, tertiary, should stock them.
It's a powerful tool for rationalizing national drug policies. Okay, we know what drugs are, their sources, names, and how they're regulated. Now, the practical bit, actually getting the drug into the patient, this starts with the dosage form.
Yes, the drug itself, the pure chemical, is rarely given directly. It's almost always formulated into a specific dosage form. This is the final product you actually administer: the tablet, the capsule, the injection solution, the ointment.
And it's not just the drug?
No, it contains the active drug plus various inactive ingredients called excipients, things like fillers to add bulk, binders to hold a tablet together, disintegrants to help it break apart, coatings, preservatives, flavorings, coloring agents. These aren't inert. They are crucial for the product's stability, performance, and acceptability.
So the dosage form provides the body, protects the drug, makes it measurable.
Exactly. It defines the dose unit, protects the drug from air, moisture, or stomach acid, masks unpleasant tastes, and controls the rate and site of drug release. Designing the right dosage form is a major part of pharmaceutical science.
Let's look at the main types. Solid forms are probably the most common, tablets especially.
Tablets are the workhorse. You have plain compressed tablets, sugar-coated ones, mostly historical now, film-coated ones, thinner, smoother. But the real innovation is in specialized tablets.
Like chewable or dispersible ones.
Yes, good for children or people who can't swallow whole tablets easily. Then you have sublingual tablets designed to dissolve under the tongue.
We mentioned this rapid absorption bypasses the liver.
Correct. Very important for certain drugs like nitroglycerin, GTN. Then there are enteric-coated tablets. These have a special coating that resists stomach acid.
Why would you need that?
Two main reasons. Either the drug itself is destroyed by stomach acid, like some antibiotics, e.g. erythromycin base, or the drug is irritating to the stomach lining, like aspirin or NSAIDs. The enteric coat ensures the tablet passes through the stomach intact and only dissolves and releases the drug once it reaches the less acidic environment of the small intestine, usually duodenum.
Clever. What about making the effect last longer? Sustained release.
Right. Sustained release, SR, or extended release, ER tablets, are engineered to release the drug slowly over a prolonged period, typically 8, 12, or even 24 hours. This reduces the number of times a day you need to take the medication, which can really improve patient compliance.
How do they achieve that slow release?
Various ways. Sometimes the drug is embedded in a matrix that dissolves slowly. Sometimes the tablet contains granules coated with different thicknesses of barrier material, so they dissolve at different rates.
Is controlled release, CR, different?
It often implies a more precise, constant rate of release, sometimes zero-order kinetics, meaning the same amount of drug is released per unit time, regardless of how much is left in the tablet. This can be achieved with more complex systems, like osmotic pumps within the tablet, Fiersol ORS system, or specialized membrane-controlled release. The goal is very smooth, predictable plasma concentrations, avoiding peaks and troughs.
What about pills? Are they the same as tablets?
Historically, yes, but the term pill is technically archaic now. Originally, it meant a small ball made by mixing the drug powder with something sticky like syrup or honey and rolling it. Tablets are made by compression. We still use the word colloquially, but pharmaceutically, it's tablets.
Okay. Capsules are another solid form.
Yes, capsules consist of a shell, usually made of gelatin, filled with the drug, which might be a powder, granules, or sometimes even a liquid in soft gelatin capsules. The shell dissolves quickly in the stomach, releasing the contents.
Can capsules be modified for release too?
Oh yes, you can have enteric-coated capsules where the shell itself or the granules inside resist stomach acid. And spancels are a type of capsule containing mixtures of drug granules with different coating thicknesses to achieve extended release.
Suppositories are for different routes.
Right. Suppositories are solid dosage forms, usually conical or bullet-shaped, designed for insertion into the rectum. They melt or dissolve at body temperature, releasing the drug for local effect or systemic absorption. Similar forms include pessaries for vaginal insertion and bougies for urethral insertion.
And lozenges.
Lozenges are like hard candies or tablets designed to dissolve slowly in the mouth. The drug is released gradually, providing a prolonged local effect on the mouth, or throat lozenges for soreness.
Okay, moving from solids to liquid forms.
Liquids are essential, especially for kids or people who can't swallow solids. The simplest are aqueous solutions, where the drug is fully dissolved in water. They usually require preservatives to prevent microbial growth.
What if the drug doesn't dissolve well in water?
Then you might use a suspension. This is a two-phase system where fine particles of the insoluble drug are dispersed uniformly throughout a liquid, usually water. Suspensions must be shaken well before each dose to ensure the drug particles are evenly distributed. Otherwise, the dose will be inaccurate.
Like some antibiotic liquids for children.
Exactly. Another type is an emulsion, which is used when you need to mix two liquids that don't normally mix, like oil and water. An emulsifying agent is added to stabilize the mixture. Milk is a natural emulsion. Again, these often need shaking.
What about elixirs and syrups?
Elixirs are clear, sweetened liquids containing both water and alcohol. Hydroalcoholic. The alcohol helps dissolve drugs that aren't very water-soluble. Syrups are concentrated aqueous solutions of sugar, or a sugar substitute. The high sugar content makes them viscous, masks unpleasant tastes, and also helps preserve them. They're very common for cough medicines.
And injections.
Injections are sterile liquid preparations, either solutions or sometimes suspensions intended for parenteral administration by needle. Sterility is paramount.
Okay. Then we have semi-solid forms, mostly for skin application, ointments, creams. What's the difference?
The base is the key difference, and it determines where they are best used. Ointments have a greasy, oily base, typically hydrocarbons like paraffin or petroleum. They are occlusive. They form a barrier on the skin that prevents water loss. This makes them good for dry, chronic, scaly skin conditions, but they feel greasy and can be messy.
And
creams are usually emulsions, often oil and water type. They contain water, spread easily, feel less greasy, and are often considered more cosmetically acceptable than ointments. They are generally better absorbed and suitable for more acute weeping lesions.
Pastes and gels.
Pastes are very thick, stiff preparations containing a high proportion of powder, like zinc oxide or starch, mixed into a base, often non-greasy. Their thickness makes them protective and good for absorbing moisture from oozing surfaces. Gels are semi-solid systems where liquid is trapped in a network structure, often appearing clear. They're typically non-greasy, water-washable, and spread easily, making them good for hairy areas or application inside the mouth, like for ulcers.
Finally, inhalations,
Yeah.
for getting drugs into the lungs.
Yes, this route is vital for respiratory diseases and also for some systemic drugs like general anesthetics. Gases, like anesthetics, or volatile liquids, which turn into gas easily, are simply inhaled via a mask or mouthpiece.
What about non-volatile drugs like asthma puffers?
For non-volatile drugs, either solids or liquids, they need to be converted into an aerosol, a fine mist or powder that can be inhaled deep into the lungs. This is done using various devices.
Right.
The most common is the metered dose inhaler, MDI, or PMDI. This pressurized canister contains the drug dissolved or suspended in a propellant, now usually hydrofluorocanes, HFAs, replacing older CFCs. Pressing the canister releases a precise metered dose as an aerosol.
Others.
Jet nebulizers use compressed air or oxygen to blast a liquid drug solution into a fine mist, which is then inhaled through a mask or mouthpiece over several minutes. Useful for larger doses or patients who can't coordinate an MDI. Dry powder inhalers, DPIs, like rotahalers or spinhalers, contain the drug as a fine powder, often in the capsule that is punctured inside the device. The patient's own breath pulls the powder out and into the lungs.
And you mentioned particle size is critical here.
Absolutely critical. For the drug to reach the target airways, the bronchi and bronchioles, the particles need to be in a very specific size range, generally between 1 and 5 micrometers.
What happens if they're too big or too small?
Particles larger than 5 millimeters tend to impact due to inertia in the back of the throat, the oropharynx, and get swallowed, not reaching the lungs. Particles smaller than about 1 millimeter are so light, they tend to stay suspended in the air and just get breathed back out again without depositing. So that 1 5-millimeter range is the sweet spot for lung deposition.
Okay, that covers the how it's packaged. Now, the how it gets in the routes of administration. Why choose one route over another?
Many factors influence this choice. It depends on the drug's properties. Is it stable in acid? Is it soluble? Is it irritating? It depends on where you need the drug to act locally or systemically. How quickly do you need the effect? Is it an emergency or routine treatment? How accurately do you need to control the dose? And critically, it depends on the patient's condition. Are they conscious? Can they swallow? Are they vomiting? Are they cooperative?
We can broadly divide routes into local and systemic. Let's start with local routes aiming for effect at the site of application with minimal absorption into the body.
Right. The goal is high concentration where you want it, with fewer side effects elsewhere. Topical application is the most obvious, applying lotions, creams, ointments, dropped directly onto the skin. Mucous membranes, like eyes, nose, mouth, vagina.
But local can also mean inside the body.
Yes. For example, giving a non-absorbable drug orally to act within the gut, like sucralfate coating an ulcer, or inhaling a drug like salbutamol, where the primary target is the bronchial smooth muscle in the lungs, even though some might get absorbed systemically.
And injections can be local too.
Definitely. Injecting a corticosteroid like hydrocortisone directly into an inflamed joint, intra-articular, aims for a local anti-inflammatory effect with slow absorption out of the joint space. Another specialized local route is intra-arterial injection, injecting directly into an artery, supplying a specific area, like giving contrast media for angiography or high-dose chemotherapy for a limb cancer. This maximizes local concentration before the drug gets diluted in the general circulation.
Okay. Now for the systemic routes, where the goal is to get the drug absorbed into the bloodstream so it can distribute throughout the body. The default is oral.
Oral administration, swallowing, is the most common, convenient, usually the safest, and generally the cheapest route. No special equipment needed. Patients can usually manage it themselves.
But it has significant limitations, which are critical to understand for mastery.
Absolutely critical. First, the onset of action is relatively slow, so it's not suitable for emergencies. Second, some drugs taste awful or can cause nausea and vomiting. Third, you can't use it if the patient is unconscious, uncooperative, or actively vomiting.
And the absorption itself can be a problem.
Yes, absorption from the gut can be variable and incomplete, affected by food, Gut motility, other drugs. Some drugs are simply destroyed by digestive enzymes, like protein drugs, for example, insulin, or by stomach acid, like penicillin G. And then there's the huge hurdle of first-pass metabolism.
We touched on this. The liver gets the first shot at the drug after absorption from the gut.
Exactly. Drug absorbed from the stomach and intestines enters the portal vein, which goes directly to the liver before the drug reaches the general circulation. The liver is packed with metabolic enzymes. If a drug is heavily metabolized by the liver on this first pass, a large fraction of the absorbed dose can be inactivated before it ever gets a chance to reach the rest of the body and exert its effect.
Examples.
Drugs like glyceryltrinitrate, GTN, lidocaine, propranolol, morphine, they undergo extensive first-pass metabolism. This means their oral bioavailability, the fraction of the dose reaching systemic circulation unchanged, is very low, and much higher doses are needed orally compared to injection, or the oral route might be completely impractical.
So how do we bypass that first pass? One way is the sublingual or buccal route.
Right. Placing a tablet under the tongue, sublingual, or in the cheek pouch, buccal, allows the drug to be absorbed directly through the oral mucosa into the veins there. These veins drain directly into the superior vena cava, completely bypassing the portal circulation and the liver.
Advantages.
Absorption is rapid effects can start within minutes, and you avoid that destructive first-pass metabolism. It's ideal for drugs needed quickly, like GTN for angina attacks.
Limitations.
Only suitable for drugs that are potent, so the dose is small, lipid-soluble enough to pass through the mucosa, and non-irritating. And the patient has to cooperate, not swallow the tablet. Buprenorphine is another drug often given this way.
What about the rectal route?
Suppositories or enemas. This is mainly used when oral administration isn't possible. The patient is unconscious, vomiting, or maybe a drug is too irritating for the stomach. It's also used for local effects in the rectum or colon.
Does it bypass the liver?
Partially. The lower part of the rectum drains via veins that go directly into the systemic circulation, inferior and middle hemorrhoidal veins bypassing the liver. But the upper part drains into the portal vein. So roughly 50% of a rectally absorbed drug might bypass the first pass, but it's variable. Absorption itself tends to be slower, less complete, and more erratic than the oral route. Plus, it can be inconvenient and embarrassing for the patient. Diazepam is sometimes given rectally to stop seizures, especially in children.
Then there's putting drugs on the skin for systemic effect, cutaneous or transdermal.
Yes, applying a drug to the skin with the intention that will be absorbed through the skin layers into the systemic circulation. This requires the drug to be highly lipid-soluble to penetrate the stratum corneum, the skin's outer barrier.
Advantages here.
Absorption is slow and sustained, providing prolonged action. And crucially, it completely bypasses the liver first-pass effect. Excellent for drugs needing steady levels over a long time.
The patch seems like the high-tech version of this, the transdermal therapeutic system, TTS.
Exactly. These adhesive patches, like for nicotine cessation, hormonal therapy, pain relief with fentanyl or GTN, are sophisticated delivery systems. They contain a reservoir of the drug, an outer backing film, and most importantly, a rate-controlling membrane.
Why is that membrane key?
Because the permeability of skin varies a lot between people and different body sites. The membrane is designed so that the rate at which the drug diffuses out of the patch is slower than the rate at which even the least permeable skin can absorb it. This means the patch, not the skin, controls the overall rate of drug delivery.
So it standardizes the dose delivery.
Precisely. It ensures a relatively constant and predictable rate of drug absorption over the lifetime of the patch, often 1 to 7 days, leading to smooth plasma concentrations, minimizing individual variation, improving compliance, just change a patch, don't need multiple pills, and avoiding first-pass metabolism.
Okay, next route. Inhalation. We discussed it for local lung effects, but also for systemic.
Yes, primarily for general anesthetics, gases or volatile liquids. The lungs have a huge surface area and massive blood flow, so absorption from the alveoli into the bloodstream is extremely rapid, almost instantaneous.
Advantages.
Very fast onset of action. Also, you can control the dose very precisely, moment to moment, by adjusting the concentration inhaled. And when inhalation stops, elimination via the lungs is also very rapid, perfect for controlling the depth of anesthesia. Some non-anesthetic drugs are being explored for systemic delivery via inhalation too.
What about the nasal route, spraying drugs into the nose?
The nasal mucosa is also quite permeable and has a rich blood supply, allowing for rapid absorption directly into the systemic circulation, again bypassing the gut and liver first pass. It's become particularly useful for some peptide drugs, which would be digested if taken orally. Examples include GnRH agonists, calcitonin for osteoporosis, desmopressin for diabetes insipidus. There's also research into using the nasal route to try and deliver drugs directly to the brain, potentially bypassing the blood-brain barrier via olfactory pathways, though that's still largely experimental.
Okay, that covers all the non-injection systemic routes. Now, the big one, parenteral administration, meaning injection.
Parenteral literally means outside the intestine, caracals beyond, enteron intestine. It refers to routes that involve injecting the drug directly into body tissues or fluids.
Main advantage.
It overcomes all the limitations of the oral route. Absorption is faster, more predictable, and often complete, 100% for IV. It's essential for drugs that are poorly absorbed or destroyed by the gut liver. It's the route of choice in emergencies when rapid action is needed, and it can be used in unconscious or vomiting patients.
Disadvantages.
Preparation must be sterile, which adds cost and complexity. The procedure itself is invasive, requires skill, can be painful, and carries risks like infection, nerve damage, or tissue injury. It's generally less convenient and more expensive than oral administration.
Let's break down the common parenteral routes. First, subcutaneous, SC or sub-Q.
This involves injecting the drug into the loose connective tissue just beneath the skin. Common sites are the upper arm, thigh, or abdomen.
Characteristics.
The subcutaneous tissue has a rich nerve supply, so irritant drugs shouldn't be given this way as it would be very painful. Blood flow is less than in muscle, so absorption is generally slower and more sustained than intramuscular injection. This can actually be an advantage for drugs like insulin or heparin, where prolonged action is desired.
Any special considerations?
Absorption relies on blood flow, so you should avoid SC injection in patients who are in shock or have peripheral circulatory collapse, as absorption would be severely impaired. There are also specialized SC techniques: needleless injection using a high-pressure dermajet, implantation of drug pellets under the skin for very long-term release, for example, hormones, or use of siolistic or biodegradable implants that release drugs slowly over months or even years.
Next, intramuscular, IM.
Injection directly into a large skeletal muscle like the deltoid, upper arm, vastus lateralis, thigh, or gluteus maximus, buttock.
How does it compare to SC?
Muscles have a richer blood supply, so absorption of aqueous solutions is generally faster and more uniform than SC. Muscles are also less richly supplied with sensory nerves, so mild irritants can sometimes be given IM. That would be too painful SC.
Depot injections.
Yes, the IM route is commonly used for depot preparations. These are formulations where the drug is dissolved in oil or prepared as an aqueous suspension of microcrystals. The drug then slowly leaches out from this muscular depot into the circulation over days, weeks, or even months, providing very prolonged action. Very useful for things like long-acting antipsychotics or hormonal contraceptives.
Any major warnings for IM?
Absolutely. Because muscles are vascular, there's a risk of hitting a blood vessel and causing a hematoma, a deep bruise. This is particularly dangerous in patients receiving anticoagulant therapy, blood thinners. IM injections should generally be avoided in these patients.
Now, the most direct route, intravenous.
Injection directly into a superficial vein, usually in the arm. The drug enters the bloodstream immediately. It can be given as a rapid bolus injection, simple push, or as a slow, continuous infusion, a drip.
Key advantages.
Immediate effect onset of action is the fastest of all routes, making it invaluable in emergencies. Bioavailability is 100% by definition. The entire dose reaches the systemic circulation. You can give large volumes of fluid if needed. Even drugs that are quite irritating can often be given IV because they are instantly diluted by the large volume of flowing blood. And for drugs with a short half-life, a continuous IV infusion allows for very precise control over the plasma concentration, titrating the dose to the patient's response.
But it sounds like the riskiest route, too.
It absolutely is. There are local risks: inflammation of the vein, thrombophlebitis, leakage of drug into surrounding tissue, extravasation, which could cause severe damage if the drug is toxic. Air embolism is a theoretical risk if air is injected.
And systemic risks.
Because the drug hits vital organs like the heart and brain at high concentrations almost immediately, any mistaken dose calculation or too rapid injection can have severe, even fatal consequences. There's no recall button. Also, a critical point: only aqueous solutions should generally be given IV. Injecting suspensions or oily solutions carries a high risk of causing embolism, blocking blood vessels downstream, which can be catastrophic. So maximum speed and control, but also maximum potential for immediate harm if not done correctly.
One last injection route, intradermal.
This is injection into the skin itself, the dermis, just below the epidermis. A very small volume is injected, raising a small bump or bleb, primarily for diagnostic purposes, like allergy testing, skin prick tests, or the tuberculin sensitivity test.
Mm.
Also used for some vaccinations like the BCG vaccine against tuberculosis, where you want a strong local immune reaction. Absorption from this site is very slow.
Wow. That covers an incredible amount of ground, from the basic definition to the complexities of delivery.
It really shows how interconnected everything is, doesn't it? Understanding the chemistry dictates the potential sources. Understanding the kinetics, the ADME, dictates the dosage form and the route. Understanding the dynamics, the mechanism informs the therapeutic use. And overlaying all of that are the crucial systems of nomenclature, regulation, and ensuring quality and access, like the essential medicines concept. It all has to work together for rational drug use.
It absolutely hammers home that using medicines effectively is a deep applied science. To wrap this up and bring it back to that professional application, let's revisit that case study scenario. A young child, 5 years old, comes in acutely ill with pneumonia. High fever, rapid pulse, breathing fast, seems irritable, and maybe a bit lethargic. The doctor needs to start an antibiotic right away.
Okay, a critical situation demanding rapid, effective treatment. Let's say the chosen antibiotic is available in 3 forms: an oral liquid, an intramuscular injection, and an intravenous preparation.
So here's the final thought for you, the listener, to really synthesize this. Considering this child's acute state, irritable, maybe difficult to cooperate, breathing rapidly suggesting severe illness, and the absolute necessity for getting effective antibiotic levels into their system fast and reliably, which route, oral, IM, or 5E, is likely the most appropriate in this emergency?
Think about the limitations we discussed. The child is acutely ill, possibly nauseous, or unable to reliably take oral medicine. Oral absorption is the slowest and potentially most variable, especially in sick children. IM offers faster, more reliable absorption than oral.
Oh.
But 5V gives you 100% bioavailability instantaneously. In a severe, potentially life-threatening infection like pneumonia in a young child, the need for certainly knowing that the full dose of antibiotic is definitely in the bloodstream immediately to fight the infection often outweighs the risks and inconvenience of starting an IV line.
So despite being the most invasive, the IV route might actually be the safest clinical choice here because it guarantees rapid, effective delivery when time is critical.
In many such acute scenarios, yes. The pharmacokinetic certainty of the IV route becomes paramount when dealing with severe illness. It's a perfect example of how mastering these fundamental principles, kinetics, routes, patient factors, directly translates into critical, potentially life-saving clinical decisions.
That really underscores the importance of mastering these fundamentals. It's not just academic, it's about patient outcomes. Thank you for joining us on this very deep dive into foundational pharmacology. We hope this detailed exploration provides that solid bedrock for understanding and applying drug science at a professional level.